Good evening, everyone. Today we are having first guest lecture. I am very pleased to introduce Dr. Pankaj Yadav. He is assistant professor in Department of Bioscience and Bioengineering at IIT Jodhpur. Today he is speaking on applications of mathematics in biology. I hand over this session to Dr. Pankaj Yadav.
Thanks, Dilpreet, for a kind introduction. So, without delay, let's start. Let me first share the screen. I hope the screen is visible.
Yes, Pankaj, it is visible.
Okay, great. So thanks. So in today's talk, I'm going to speak about application of logistic regression models to investigate gene-environment interactions. And let me start with the outline of my talk. So I will first introduce about the concepts in gene-environment interaction studies. So what are different ways people understand about these gene-environment interaction, and then I will focus on case-only design, which is a kind of non-traditional study designs to investigate gene-environment interactions. And then I will show you some applications of this approach that is used for studying the case only design in gene environment interaction studies. For that, I will elaborate 3 projects. And in the end, I will summarize the talk with some interesting findings from these projects. So let me start with the first different ways people understand the gene environment interactions, because biologists have a different understanding of this concept, and also there are different ways that is described in the field of mathematics and statistics, how this is understood. So in this gene environment interaction, we have environmental inputs, like we have alcohol, smoking, or what kind of lifestyle we are following, and what we are eating, where we are working. So these all composed as an environmental inputs. So this is, we can say, the environmental profile of individuals. And then we have the genetic makeup of the individuals, which is constant and also inherited from families or from parents. So, so it is now, and also now it has been observed for long that these 2 factors, the genetic factor, the genes, and environment interact and somehow to influence the disease conditions in individuals. So these environmental factors, if you look at the metabolic level, they kind of try to change the responses or try to modify the metabolic networks, or somehow try to change the pathways that are involved in the diseases that occurs in individuals. So this is like a broader description of this concept and why it is so important. So this is cartoon that I really like. So once you have identified a list of genes that might be interacting with environment, so somebody might be interested in knowing that do these genes, if I have these genes, can make me look fat. So this depends upon like whether these genes are interacting with the environment or not. So this cartoon is basically is depicting that point. So let's now jump to a more statistical or mathematical understanding of this concept. So in genetics or in biology, usually we have two factors. We have disease as one factor, which is our, you can say, the response. And then we have the many predictors, like genetic factors. So usually traditionally what people have been doing in this field is that they were looking for association between the genetic factors and with the disease of interest. So this is basically done with the different association tests that are available. And now a very non-traditional kind of or advanced way of understanding the diseases is now that we are including nowadays environmental factors also in studies. So basically what this picture, this figure is showing is that this association is influenced by the presence or absence of the environmental factor like smoking. So if this factor is changing this effect size, you can say, then we can say that this environmental factor is interacting with the genetic factor in causing the disease. So in one line, we can say that we can describe G cross E, gene environment interaction, as the modification of the genetic effect by the environment. If we see this modification, then there is a gene by interaction. I hope this is clear now a bit, or at least obvious. So I give you one example of a very recent, but about few years back. They have identified this gene environment interaction in ulcerative colitis disease. So what they have found is that they found an interaction between smoking, and there is a gene called CALM3 gene, which is which has been associated with ulcerative colitis disease. And what they have found in their studies is that they have two groups, let's say smokers, and they have non-smoker groups. So what they have found is that they found that in smoker groups, they have the low genetic risk, means that for the same variant of this gene. So in biology, we can have different forms of a same gene that are called alleles. So for the same allele, the smokers have low risk, whereas as the non-smokers have the high risk. And this might be a bit funny also, because smoking is kind of trying to reduce the risk of this ulcerative colitis disease, but this doesn't mean that smoking is good for health, because there are other serious diseases that can happen, such as lung cancer, that can happen with the smoking. So also this study was not the only one, but there were many other studies that followed, and also previously some people have already found that smoking has a protective effect on this ulcerative colitis disease. But the biological mechanism is that not much that well understood, but there are several studies that have shown the same thing. Okay, so this is just telling us this why gene environment interactions are important. So some people might be exposed to some kind of environment, though they have the same genetic profile, but some might be exposed to different environment to give a different risk for some certain diseases. So in this study, they have shown that the genetic risk depends upon the environmental exposure. Now, how we study these kind of interactions. So we have in literature, there are several types of study designs that are available for studying gene environment interactions. So traditionally, there has been existing case control studies where we have a set of two groups of individuals, which are cases and controls. Cases are the people who are affected with the disease, and the controls are healthy individuals. So this is one way people have been using it. And then there is a case-only design, which also got high interest because of its more statistical power, and also its practicability of collecting the data. So here we have to collect the data on both cases and controls, but here we only need the data on the cases to infer the gene environment interactions. Also, there are some family-based designs like trios, where also information on the genetic profile of the parents is needed, and besides the genetic data of the affected individuals. So these kind of family-based designs, they restrict us because collecting data sometimes might not be feasible for the parents, because some diseases might occur at a late onset, like this is also the case, I think, for most of the environmental related diseases, where the availability of the parents not be possible. So today I'm going to focus on this case only design because of its practicability to collect the data, and also it offers more statistical power. So so let's first take a look how mathematically, or how this is done in case of case control design. So as I said, this is a more traditional way of studying gene environment interactions. So let us assume that we have three factors. Let's say D is denoting the disease status of the individuals. So 0 means the disease is absent, and 1 means the disease is present. So for simplicity, we also consider that the genetic factor is present or absent or present, let's say. And then we have also environmental factor. Let's say we also, for simplicity, consider it as to be absent and present. 0 means the smoking factor is absent. So in case of smoking and non-smoking, it can be like non-smokers and smokers. So we have these three factors, and then we have, as most of us are aware of, this logistic regression model that we can that has been deployed for studying this the interactions and how. So we have in this model disease status as our response variable, and then we have different coefficients that are denoted by betas. So beta 0, beta g, and beta e and gamma are the coefficients. And we have a G and E denoting the factors, variables. And also we have an interaction term between these two factors. So gamma is actually giving us the estimate of the interaction between these two factors, genetic and the environmental factor. So this is the model that has been used in when people use case control designs. They fit the genetic and environmental data of the cases and controls to this model, and then they estimate the gamma. That gives us the estimate of the interaction between these two factors. And the beauty of this model is that these coefficients like beta g and beta e, they can be expressed if we take the exponential of these factors, they can be expressed in terms of the odds ratios. And as we know, if you have read some papers in this field, that most of the effect sizes that are reported in publications is odds ratios and also the relative risk, but odds ratio is more frequently understood and also very well acknowledged in the publications. So for example, here we have, if we take the exponential of beta g, that give us the odds ratio for the genetic factor. So basically ORG here is the disease odds of individuals which have only the genetic factors relative to those individuals which do not have any of the factors. So that gives us the odds ratio for the genetic factor. Similarly, we can define ORE and ORGE. So which is giving us the so by just doing some manipulations, we can estimate the gamma from this expression. All right, so this is the case control traditional way of studying the gene environment interactions. Now I want to show you this case only design. So in short, I don't know how to remove this panel. I hope. Okay. Okay, so we have learned about the case control. Now the next that I want to show you is the case only design. So here we have some assumptions that are to be met before we can proceed with the uses of this study design. So the assumptions are very important. So first assumption is that G and E factors, so genetic and environmental factors, have to be independent in the general population. So which implies that the genetic profile of an individual should not determine the environmental profile, which is true in most of the cases, right? Our environmental exposure is not determined by our genetic profile. So this assumption seems very reasonable. So most of the time this can be satisfied. And other assumption is that the disease has to be rare. So this means a lot of simulation studies have been done, and rare means that here, if we look at the simulation results, if we have a prevalence of the disease greater than 5%, then there seems to be some bias observed then in the estimates from this study design. So there is this paper, Pigos, in 1994, they actually proposed this case-only design. But later it did not receive much attention because people got attracted towards something else. They just were thinking that only the genetics can explain most of the diseases, but over the years it could not happen, and now the people have now started realizing that environmental is also an important factor that need to be considered in when doing such kind of analysis. So Pigos, they have given a mathematical explanation and also a mathematical proof that this case only design, the estimate, if these two assumptions are satisfied, then the estimates from this case only design is equivalent to estimate that we get from the case control approach. So what I want to say is this. So the estimate of this gamma is equivalent to the estimate that we will get from the model that we will be using for the case only design. So in the follow-up slide, I will show you the model. So before that, I just want to say that if these assumptions are satisfied, then we don't need controls, and we just use the data on the cases itself to infer the existing gene environment interaction. So we just basically then look for association between these two factors, G and E, in the cases. If we find that there is an association, that will point towards the gene G cross E at the population level. So this is the main point I hope it is I am able to convey it, that we need to only look for an association between G and E if these assumptions are satisfied, and if we see that this association is significant, then this will point towards the G cross E at the population level. So now coming back to the model that is employed in here in this case only design. So how we do the testing here? So here we have this reduced model, we can say. So we fit the G and E data here of the cases. So we don't need controls. I'm emphasizing it again, that we need only the data on cases. And then we fit this reduced logistic regression model that is shown here. So here we can we have environmental as our response, and then genetic as our predictor, or we can do the other way around. But here the estimate, this theta estimate, so the estimate of this theta is equivalent to the gamma that we have in the full model under these assumptions. So this proof is given in this Pigos paper. So this proof is a bit longer, so considering the time that is allowed. So, but if you are interested in looking at the theoretical proof of this, then you can look into this Pigos paper. It will give you a nice explanation why the theta is equivalent, estimate of the theta is equivalent to the estimate of the gamma. But just you have to just bear with me that this is true under the assumptions are, if the assumptions are satisfied. So, so as we do in most of the cases, we can now do the testing by checking whether this theta is equals to 0 or not. So that will be our null hypothesis. And this is also actually equivalent to testing gamma equals to 0 in the full model. So we can use a classical Wald test for that. And the estimate of Wald statistics can be used here to get and for the test of this whether there is an interaction, whether there is an association between these two factors or not. If there is an association, means there is also an interaction at the population level between these two factors. So we can check that by taking a normal distribution. And then I want to show you some applications of this case-only approach in the field of genetics. How we can use these case-only studies. So the first example I want to show you is in the role of linkage disequilibrium in case-only studies of G cross E. I know some of the terms might be weird for you, but yeah, believe me, I will be able to explain them to you. So yeah, so here we have this linkage disequilibrium concept. So let me first introduce you to this concept of linkage disequilibrium. It is a very well established phenomenon in genetics. So basically, it's a non-random association at a given in a given population. So it's a population level phenomenon. So it's a non-random association between the alleles. Alleles are basically different forms of the same gene. So if we have a gene 1, the gene 1 may have a different form in different individuals. So it can be gene 1, gene 2. Some individuals might have gene 1, some individuals might have gene 2 at the same locus or same location in their genetic code. So these are called as, so gene 1 and gene 2 are called refer as alleles. So so we can have, likewise we can have different genes in these individuals. So basically linkage disequilibrium is a non-random association between the alleles of two or more genetic loci. So we can have different alleles at different locus. So if this is basically this is a consequence of basically a physical linkage between genetic loci. So during the biological recombination and when we have cells are dividing, and when we have the transfer of information from parents to the child, then this kind of information is conserved. So basically it is a consequence of physical linkage, but it's not equivalent to, means that sometimes it is also seen that there is there is an association between far appearing alleles on different that are separated by very large distance physically on the genetic or on the DNA, you can say. So it has many applications. So one application is in genome-wide association studies, very well known as GWAS in field of statistical genetics. And their application is not, so the application of this LD is not known in the field of gene environment interaction studies. And particularly using the case-only approach, it has nothing was studied. So in this application, I want to show you how this can be this application of this LD can be useful in studying the G cross E interactions, particularly using the case-only design. So let me again explain you a little bit more of biology here. So I hope you're familiar with the chromosome of definition. So you can consider DNA. You can consider as a book. If you look at the whole DNA of an individual, you can consider it as a book, and the chapters you can consider as a different chromosomes. So even if in the chromosome we have, if you look, so this basically is showing like very much abstractive view of this chromosome. So we have, let's assuming that this is our chromosome, then we have, as I said, we have different genes. So there is a concept called polymorphism. So different polymorphisms actually can happen in the genes depending due to the as I said, due to recombinations and some biological phenomena. So let's say we have here gene 1, and here we have gene 2. So we have I'm calling it as SNP1 and SNP2. Basically, these are you can say the locations within the gene. So one you can say the nucleotide or single base positions, you can say. And so what is LD actually is how these associations. So since these are polymorphisms, so some individuals might have, let's say here, as we know that DNA is composed of 4 bases, A, C, T, G, right? So some individuals might have here in this position A, some individuals might have T. So this is called polymorphism. But if all the individuals have just A at this position, this will not be considered as a polymorphism. So polymorphisms are like variations in the single nucleotide positions. So as I said, if some individual, I'm just giving an example. If some individuals have A and here some individuals might have T, whereas in this position, some individuals might have a C, and whereas some individuals might have a G here. So if we see that some if there is some association between C and A here, some non-random association, like most of the individuals, they always see that there is A and then there is a C, then we can say that okay, there is something going on here. So there might be some disequilibrium here between these two. So there might be some non-random association between these two SNPs. And now I want to talk about the interaction. So let's say we have another SNP, or we can say SNP. You can as consider. I mean, I know it's difficult to understand, but you can consider it as that still it is a gene. So we can say it is a gene, and some, as I said, there are different forms of the gene. So some individuals might have one copy of the one form of the gene, other individuals might have different form of the gene. So this, if let's say we assume that this is interacting, so let's say this gene is interacting with some environment, so this we assume that this is happening. Now the question comes, can we use the neighboring genes or SNPs to infer the underlying interaction effect, given that the SNP star is missing in our data? So this can happen due to several reasons. Like this can happen due to quality control, or might be some technical issues that this region is not sequenced or genotyped by the machine. Now the question here, the research question here is that, can we use, in the absence of SNP star, can we use SNP1 or SNP2 to infer the underlying effect, interaction effect? So can we use SNP1 and SNP2 as a proxy for SNP star? That is the question. Yeah, so for this, I have done some simulation work. So as I said, I have used this case-only approach. So what I did is, just to explain you that what I did is I simulated the whole population, though, using the full logistic regression model with the, because we have to simulate the whole population, right? So we cannot just simulate the cases here with the help of this model. So what I did is I simulated the whole population with these parameters. So I have used different parameters, but then based on some settings, it was found that these parameters were optimum for to also to make the assumption of the case only design to be satisfied for the rare disease. So here I have considered a moderate genetic effect in my simulation. So and also I use different interaction effects to simulate the data. So the data was simulated, though I simulated the whole population, but what I did is I only randomly picked, or I simulated until the population until the desired number of cases that has been reached. So here both cases and controls were simulated, but let's say if I remember correctly, I used 1,000 cases. So until the 1,000 cases were obtained, the simulation was run. So I have used different disease models. So we have in genetics, we have additive model and dominance model. So we can have like different settings for this kind of model. So based on how the genes are, or the alleles are affecting the disease, like we can have different copies of the same allele in the individual. So usually we have two copies of the one allele in the one individual. So this was the simulation setting for this purpose, and then I want to show you some results from the simulation. It's quite interesting results. So as you see that here we have on the horizontal axis we have the LD. So just for simulation, actually we don't need two proxies. We just simulated one proxy. So if we establish that it works for one proxy, then it can be extended for the other proxy SNPs also. So I simulated one proxy SNP, and then I simulated, as I said, 1,000 cases from that full model, and I use around 5,000 replicates to estimate the statistical power. And blue and black color here corresponds to two different models. The blue one is dominance, the black one is additive. So here on this horizontal axis we have the LD. So LD between what and what? LD between SNP star and the proxy SNP. So this LD can be varied. Actually, it's I just want to say that here the measure that is used here is the Pearson's correlation between the two SNPs. And as I said, this can have vary the range from 0 to 1. So 1 means there is a perfect correlation between the two SNPs. So if there is a perfect correlation or perfect LD between the proxy SNP and the interacting SNP, then we can have a value of 1, and if they are independent, then we have a value of 0. So these were also simulation settings. So simulations were changed so that we get different levels of the LD between these two interacting and the proxy SNP. Okay, so on the vertical axis, what we see is the statistical power to infer or to infer the gene environment interaction at the proxy SNP. Remember, we are interested in estimating we are interested in seeing whether proxy SNP can be used to infer the underlying interaction or not. We have introduced the interaction in our data with the help of, as I said, in the simulation setting. I am going back one slide. So I have already shown that we have introduced the interaction. Now we want to see whether we are able to estimate this interaction or not from the proxy SNP at the proxy SNP. So as you can see that there is a non-linear relationship between the statistical power that we see and the amount of LD that is between the proxy and the interacting SNP. So you can see that when they are in perfect LD, then we get almost we get close to almost power of 1. So this points towards the fact that now we can say that we can power to detect that interaction is an increasing nonlinear function. And when we have also seen that the additive model is giving more power compared to dominance model, that is another observation from this study. Now, also we have shown that I've used different measures of the interaction. So as the interaction becomes stronger, it is easier to detect even with the dominance model. So yeah, so the gamma of -2 actually refers to that we can have, as I said, that we can have different kinds of interaction. We can have synergistic interaction, like when we have both happening in the same direction. We can have antagonistic interaction. So gamma -2 refers to antagonistic regression interaction between these two factors. And simulation results also showed that this antagonistic interaction was easier to detect, as it is visible here, that even at the lower level of LD, it was easier to detect. So conclusion from this study is that LD can be exploited to indirectly detect the underlying G cross E, and the greater power emerged under an additive model than under a dominance model. And power was positively related to the underlying interaction effect, as we saw that if we have as we increase the interaction effect, it was easier to detect it. And antagonistic interactions were easier to detect compared to synergistic interactions of the same magnitude. Now, another interesting application of this case only approach to detect interaction is in the case of population stratification. So here we have, I will show you how population stratification can affect case only studies, and how we can rectify it using some measures such as genomic control. So in case of gene environment interaction studies, we can have different kinds of stratifications. So we can have just environmental stratification. So what these bars are showing here, the two colors are representing, so the light and the dark, they are representing two different populations, you can say Asian and European populations, let's say. So if these two populations have just a difference in the environmental frequency, let's say Europeans might have a different smoking frequency compared to Asians. I'm just giving an example. I don't know what's the reality, but let's say that they have higher smoking frequency compared to Asians. So we can say that these two populations are stratified by environmental factor. And this is one scenario. Then we can have, so FG and FE basically refers to risk allele frequency and exposure frequency. So we can also have another scenario where the two populations are just stratified by genetic factors. So in this case, these two populations will have only the differences in their genetic risk allele frequency. Okay, so we can even have a more complex and even, I think, practically more relevant, or you can say that is more frequently seen in practice, that both genetic and environmental factors are stratifying the two populations. So again, for this, I have done a couple of simulations to simulate these different scenarios of stratifications. Again, I have deployed this case-only approach, and that model that I showed you in the beginning, the full logistic regression model, that is to simulate the data. And here I am showing you some settings that I have used. So so delta FG is the risk allele frequency difference between two populations. Delta FB refers to exposure frequency difference between the two populations. So I have used different settings for this. For, let's say, for creating a PS for creating genetic stratification, I have set a PSE equals to 0, then we can have different values of delta FE to create the environmental stratification. Similarly, we can have a PSG scenario where we only change the delta FG, where we whereas kept delta FE to as same in both the populations. So, and in the third scenario, I have varied both delta FG and delta FE to create the different scenarios of the joint stratification by the two factors. And just to point that interaction term was kept same in both populations. So I assume that the effect of interaction in the two populations is in the two subpopulations is same in the two settings, in the different settings. So here I want to show you some results from the type 1 error rate. So how we have shown for the setting where we have no interaction. So let's say because we want to see what is the type 1 error rate, so we just we have introduced no interaction. So in reality there is no interaction in the data. And then I looked at environmental stratification setting and looked at how the type 1 error rate is behaving in this under this kind of stratification. So for this, as I said, delta FG was kept constant, and that is 0, but whereas the delta FVE was varied from 0 to 0.4. But you can see that this does not affect the validity of the case only approach. So again, I'm just to refresh you that for here I'm using for estimation, I'm using that reduced model that I've shown for the case only design. So the conclusion you can say here is that there is no effect of environmental stratification on type 1 error rate. So and also this was same thing was observed in the same phenomenon was observed under the genetic stratification. So there is no effect on type 1 error rate under different settings of the genetic stratification. But when we have the joint stratification, then we are in trouble. So you can see here the results. So here you can see that we have different levels of delta FG, that is different levels of genetic stratification, and then in different colors showing you the different levels of environmental stratification. So delta FE equals to 0, which is this line. Here we just have the genetic stratification because delta FE was kept 0 here in these settings. But as we increase delta FE, then we see that this that inflation is exponential, you can say. So we see that the type 1 error rate can be close to 0.9 when we have a setting of where we have the genetic stratification equals to 0.4, whereas and environmental stratification is 0.10. That is 10% difference between the two subpopulations. So now this is an important issue also, because most of the time we get the data that is sometimes you are not aware of whether it is coming from a single population or it is coming from different population. So this kind of stratification can give us false positive results. So we must to be careful. So now what are the approaches to deal with this? So there are different ways to account for this population stratification. So the people have proposed different ways. So Pritchard has in 2000 has proposed a stratified analysis. Then so here one can separate the stratas and then do the analysis on different stratas. So that is one way, and then we have another approach in genetics that is called as genomic control. That was also very popular. That was proposed in by Devlin and Reuter in 1999. And then there was also a transmission disequilibrium test, TDT, popularly known as, that was also proposed in this along the same year. But that has not been used. So, but we don't know whether it works or not for the case only. So, but this has been proposed in the context of some other studies, that is case control or other different study designs. For example, TDT is proposed for trios study design. So, what I did is I looked at different way of counting the stratification in the paradigm of case only design, and looked whether these strategies might work or not. So as I said, in this stratified analysis, what I did is I included the subpopulation membership variable X, let's say, in the case only regression model. So as I showed you previously, that this was the model that we showed previously in the slides back. So now we have another variable, that is a subpopulation membership variable, that is added to this model. And now the significance of this G cross E is assessed by using a test statistic, that is I call it as W strat, because we have now a stratified measure of the estimate. Now this approach assumes that population affiliation is known in advance, but this is not possible in practically in most of the data analysis scenarios, where we don't know whether which sample comes from which population. It does not happen always. Maybe in some cases, but not always. So this doesn't seems to be more. This approach doesn't seems to be more. Does not seems to be more fruitful. So another approach is genomic control, where we use the idea of a modified test statistics of interest using a moderate number of markers, let's say M markers, that are unlikely to be disease associated themselves. So here the idea is that we need to have some prior knowledge that this region of the DNA is nothing to do has nothing to do with that with the disease of interest. If we have that kind of information, then we can use those kind of genes or SNPs as markers, and we can estimate an inflation factor from those markers. Again, this inflammation factor is sum of square of the Walsh test statistics of those markers, and then we kind of take the mean of those test statistics. So WI here is the, as I said, the Walsh test statistic and for the ith marker, and then we use this inflation factor to correct for the issue of population stratification. So here we use at the test marker, so at the marker interacting marker, we use the modified test statistics that is corrected by the inflation factor. So yeah, this has been shown to have a F distribution with the 1 and M degrees of freedom. So this in this approach, it is now it is not required that we have to have any information on population affiliation or not. We can always use this approach even if we don't have such kind of information. And TDT I am going to skip because it is basically used for some other kind of designs, but just to show you that this is also one approach for correcting for the population stratification issues. So now again I did some simulation work to show you that how these different measures of correction, population stratification correction works. So under this setting, I'm going to show you how we have used this genomic control approach. So and in the genomic control approach, I have simulated 500 markers independent and that has nothing to do with the disease of interest. And then for that, I have picked different different frequencies for the risk allele of these 500 markers. The frequency was in this range for this. And for trios data, I'm not going to talk, sorry, because I'm also kind of exceeding the allowed time. So I just want to show you some interesting results here. So here we have, I'm going to show you first the type 1 error rates. So quickly, so we don't have any interaction in the data. Then we looked, we changed the delta FE to different levels. So I just multiplied it by 100 for simplicity. So we have already a different, we have already a joint stratification here. So this was the traditional design, traditional study design, case only design statistics. So here you can see that if we increase the delta FE value, we see the inflation in the type 1 error rate. And then we have WGC, which is correct test statistics corrected by the genomic control approach. So here we can see that the type 1 error is controlled, but as the level of joint stratification increases, it becomes more conservative as the type 1 error rate goes below 5%. And I also looked at WSTRAT, that is stratified analysis. That seems to be working very well, but it's only slightly conservative, but still working very well. But as I said, this might have a difficulty because we don't know sometimes the affiliation of the population individuals. So we also looked at TDT, that also worked well. So finally, I want to show you just, I think this is the last slide I want to show you. So this is the power, statistical power that we get under different settings of the interaction effect. As you can see that, let's say for this interactions effect, you can see that for the GC we see a reduction in the power also when the interaction, the level of stratification increases, but at moderate or very small level of joint stratification, we still get a good power compared to surprisingly, even we are getting more power compared to WSTRAT and TDT. But if this was very like you can say very low level of the interaction, we have where gamma equals to 0.5 corresponds to a moderate interaction. So for a moderate level of interaction, we get good statistical power in all the test statistics, even for the WGC. But the trend is still there. As you can see, when the level of interaction stratification is higher, then there is a drop in the statistical power. All right, so I think I will skip this part. So yeah, so in summary, I have shown you two things. One is that we can use LD information to indirectly detect the underlying G-cross-E interactions by following a case-only approach. So the proxy SNPs can be used with the help of which we can infer the underlying interaction if there is any interaction. And I also shown you that genomic control approach can be employed to correct for population stratification issues in case-only studies. And then I skipped that, but that was like more applied statistics or applied application of this on this inflammatory bowel disease, where I identified some interactions between gene and smoking for different countries. So if you're interested, I will definitely be able to explain you, and if you're interested, you can approach me later. So with this, I would like to thank all those peoples that have contributed to this work and who has helped me in providing the data, and also helped me in providing the statistical guidance. So yeah, thank you. And if you have any questions, please let me know. So with this, I am closing from my side and happy
Thank you,
to take
Dr.
the
Yadav.
questions.
Thank you, Dr. Yadav. Okay, so participant, do you have any question for Dr. Pankaj Yadav? Hello. I have unmuted everyone. You can unmute yourself and interact with Pankaj Yadav. I think there is no question for you, Pankaj.
I hope I was able to deliver some messages, though I know it's more complicated because of the involvement of biology concepts.
Yes.
Yeah. But you can always write me back.
Yes. Somebody wanted to ask something? Okay. But I find this talk interesting. However, I also understood only partially, but it was interesting applications of regression in biology. Okay,
Okay,
thank you,
thank
Doctor.
you.
Thank you, Doctor. So, thank you very much for delivering this wonderful talk. Thank you.
Okay, my pleasure. Thank you for inviting. So, shall I close?
Yes, thank you.
Okay.
Okay, bye. So participants, tomorrow we will continue with our discussions on linear algebra. There will be a lecture by Dr. Kiran Hirimat at 5:30 tomorrow. Okay? We will be meeting tomorrow at 5:30. See you. Bye.
Okay, ma'am. Thank you.
Bye.